Hi, everyone. Welcome to our Friday Noon Talk. It's my pleasure to introduce today's Noon Talk speaker, who's Marlene Kontcho. So Marlene obtained her bachelor degree in biochemistry and a master's degree in applied molecular biology from the University of Maryland, Baltimore County. She worked in the pharmaceutical industry in upstream and downstream monoclonal antibodies manufacturing and process improvement before she started working as a research technologist in the radiology and health branch at Canadian Nuclear Laboratories in Chalk River, Ontario, where she is researching low and high doses radiation effects on biological systems. And she's working with our colleague, Dr. Richard Richardson, studying how the oxygen effect is impacting the development of radiation-induced cataracts, which indeed is the topic of her presentation today. So I'm going to hand over to you, Marilyn, and thank you very much.
Hello, everyone. I hope you all have a great week. I would like to thank you for the nice introduction.
Are you able to share your screen, Marilyn?
I'm doing that right now.
Perfect.
Can you guys see my screen?
Yes.
All right, great. Okay. So the presentation I prepared for you today is on the investigation of the oxygen effect on the irradiation of lens epithelial cells and its possible role in cataract formation. So this talk would be more about presenting the project, its relevance, and I will also show some preliminary results that we collected so far. A brief overview of the presentation. I will talk a little bit about the human eye lens and lens epithelial cells, the different type of cataract, and the link to ionizing radiation and why it is relevant to study them. The concept of the radiation oxygen effect and what is currently known about it. Then I will present the cataract project or goal and how we're studying the oxygen effect in lens epithelial cells. And finally, I will go over what we're currently doing in the lab and what we're planning on doing in the future. So the human eye lens, it's located just behind the cornea and the iris. And as you can see in the top picture, it is avascular, which means that it's lacking blood vessel. And it's surrounded by the aqueous and the vitreous humors, from which it gets its nutrient and oxygen. It's enclosed by a basal membrane, also called capsule. It has a nucleus in the center, cortex, and a layer of epithelial cells on its interior face on the cornea side. The lens play a very important role in the vision process. By changing its shape, the lens can focus light rays passing through it onto the retina, which allow us to create clear images of objects. The formation of the lens starts in the embryo, with the nucleus forming during week 4 to 5 after fertilization. It is the only tissue in the body that grows throughout life, with the layer of these very long concentric lens fiber cells that can reach up to 12 millimeters long, stacking on top of all the ones as they form. These lens of fiber cells are conserved, they accumulate and do not divide. And with age, the protein that they contain will unfold and aggregate, turning the nucleus yellow. The lens is also known to be one of the most radiosensitive tissue in the body and can sustain damage when exposed to lower dose of radiation compared to the rest of the body. So like I mentioned before, the anterior face of the lens is covered by a single layer of cuboidal epithelial cells, also called lens epithelial cells. I will be referring to them as helices from now on. They are responsible for the lens growth and most of the lens metabolic activity. So you can see on the figure the pumps and the movement of ions, amino acid, and antioxidant, especially glutathione, which is a natural cellular antioxidant. So remember that the lens cells are retained for life, and they are very rich in protein that are susceptible to damage by oxidation, which accumulate with age. It can be detrimental to the function of the lens. Therefore, they need to be very well protected. The lens rely on this transport to get this nutrient and antioxidant from its surrounding, including the iris, the retina, the ciliary body, the aqueous and vitreous humors. And some of these processes require energy, which helices provide. Helices are also divided into zones. So if you look at the picture, you have the central zone. These cells do not divide much, but they're responsible for most of the lens metabolic activity. Then on top and on the bottom of the central zone, you have the germinative zone, and these cells are mainly responsible for lens growth. They divide throughout life, although the rate of division slows down with age. They divide and differentiate into lens fiber cells as they migrate from the germinative zone to the transitional zone and the meridional zone at the equator. And as they move, and when they get there, they elongate and lay on top of all the fiber cells. So during this process, they lose the organelles to become mature lens fiber cells. So they lose the nucleus, they lose the mitochondria, and they significantly increase the content of highly organized crystalline proteins, giving the lens its transparent look. So if you can just note where the nucleus, the cortex, and the periphery of the lens are looking on this graph, it will help you a little bit understand the different type of cataracts, which I will talk about in the next couple of slides. Cataract is the cloudiness of the lens, which is normally transparent. It is the second overall cause of visual impairment, the first one being uncorrected refractive errors, and the major cause of blindness if left untreated. Cataract are normally associated with aging, but it's also linked to genetic and metabolic diseases such as diabetes and asthma. And there are three major types of cataract. So you can refer to the picture on the right. It will give you a visual representation of each of them. So starting with nuclear cataract in figure A, which is the most common type of cataract and affects the nucleus or the central portion of the lens. Then we have in figure B, posterior sub-capsular cataract with opacity in the back of the lens near the capsule. And finally, we have cortical cataract in C, which are recognized by the spoke-like opacity, which extend through the cortex of the lens to the periphery of the lens. And also you can observe more than one type of cataract in a single patient. The onset of specific type of cataract, it's facilitated by some risk factors. So in this table, you can see a list of common risk factors found in the literature that are known to increase the risk of cataract onset. Single or double arrows means that the cataract type is moderately or strongly associated with that risk factor. So nuclear, but also cortical cataracts are highly linked to aging, which is the most common cause of cataract, while posterior subcapsular cataract or PSCs, unlike the other type of cataract, has a very high prevalence in atopy, calcium imbalance, ionizing radiation, which will be the focus of this presentation, ocular inflammation, and steroids. So those steroids which are used in the topical treatment of ocular inflammation, and as well as retinal dystrophy. So other risk factors for PSCs are diabetes and vitrectomy, which is the surgery to remove the vitreous humor. But then also not shown in the table, but important to highlight, it's that nuclear cataracts are highly linked to congenital and hereditary disease, as well as high oxygen levels and hyperbaric chamber treatment, while PSCs prevail in low oxygen levels in diabetic patients. So as of today, cataracts are listed as a deterministic effect of radiation rather than a stochastic effect. Deterministic effects such as skin reaction are observed when a threshold is reached, which is a dose high enough to cause cell death and then injure the tissue. Whereas stochastic effects such as cancer follow the no threshold linear model, which means that there is not really a threshold required, but the probability of the condition happening increases with the dose received. Before 2011, the International Commission of Radiological Protection set the threshold for detectable cataract to 2 grades for acute exposure and 5 grades for fractionated or protracted exposure. However, the many study completed on animal lens model and epidemiologic review on astronaut, radiological workers, medical staff, and atomic bomb survivor that showed the onset of cataract could happen at lower dose than the one suggested by the ICRP, which prompted the organization to amend the cataract threshold limit several times. And that threshold was significantly lower to 0.5 gray of radiation exposure in 2011, with an occupational dose limit of 20 millisievert per year over 5 years, with no single year exceeding 50 millisievert. Other epidemiologic studies in industrial radiographer, as well as looking at the mechanisms, the biological mechanism of radiation-induced damage on human lens and animal lenses, shows a strong correlation between late cataract onset and radiation doses as low as 0.1 gray. So this dose may not necessarily lead to cell death, but then they can cause enough DNA damage and generate and create oxidative stress that accumulates and, if given enough time, can develop into cataract. This damage can lead to a process that we call epithelial to mesenchymal transition. I will talk a little bit more about that later on, which is characteristic which is pretty common in malignant cancer, in cancer being a stochastic effect of radiation. All these findings are challenging the ICRP recommendation, which might be an overestimation of the real threshold limit for cataract. And it opened a debate for cataract to be reclassified as a stochastic effect without any threshold, and showed the need for more studies to determine the mechanisms for radiation-induced cataract. So why is the study of radiation-induced cataract important? Well, there are many people at risk, and some of them do essential work for us, which is why it's very critical to establish the proper guidelines and protective measures to mitigate this risk. The information that you see on this slide are adapted from a paper by Boufflet and all in 2012. It is a memorandum from the Health Protection Agency responding to the 2011 ICRP new guidelines for cataract and lens protection. I will be using the term a risk ratio, or RR, which is the probability of developing a condition when exposed to a specific risk factor. Starting from space radiation on the left going clockwise. So we have astronauts and air pilots that are exposed to space radiation. We're talking about high and low linear energy transfer radiation, including high energy protons, heavy ions, gamma and X-ray. And there's a risk ratio of 2.32 for cataract onset for astronauts exposed to a dose higher than 8 millisievert, while pilots have a risk ratio of 3.02 at 1 gray. We have, for radiotherapy, we have people undergoing radiotherapy treatments, such as childhood cancer survivors and patients that need CT scan, multiple CT scan, especially for the diagnostic of head and neck cancers. And they have a risk ratio of 3.2 at doses higher than 2 gray. The people most at risk will be the radiological workers, especially the medical worker, with occupational cardiologists having a risk ratio of 5.6, which is pretty high at 1 gray, and radiological staff a risk ratio of 4.6 at very low dose with no apparent threshold. And let's not forget about nuclear incident survivors that were exposed to gamma ray. The risk ratio is quite low compared to the others, but it's still existing. So a proposed mechanism for the development of posterior sub-capsular cataract, it's that the exposure to ionizing radiation induces oxidative stress and disruption of the ion pumps in the lens, which lead to the aberrant proliferation and differentiation of LECs. So if you follow the red arrow on the figure, and so it will help you understand a little bit more. So instead of differentiating into elongated lens fiber cells as they move towards the equator of the lens, LECs instead become these bladder-like cells that accumulate at the back of the lens via a process that we call epithelial to mesenchymal transition, during which LECs will lose the polarity as epithelial cells and also the cell-to-cell junction, and gain migratory mesenchymal phenotype. Then they get to the back of the lens, they accumulate, they can get inflamed, lose the osmotic balance, and they become necrotic and swollen. They break down into vacuoles or dense opacity that grow in size with time, resulting in cataracts. Additionally, the sustained inflammation of extralenticular tissue that are close in proximity to the lens, such as the uvea, which is the middle layer in red that you see in that eye diagram on the top. And that includes the choroid, the iris, the ciliary body, as a result of radiation exposure or other risk factor, has also been linked to the onset of posterior subcapsular cataract, which means that LECs do not have to be directly hit for cataract to form. And we can see that with astronauts suffering from spaceflight associated neuroocular syndrome due to prolonged microgravity exposure in space, or patient with retinitis pigmentosa, which is the breakdown of cells in the retina. And there is a 92% cataract incidence in high-dose treatment of choroid cancer. And this risk is even higher for anterior tumors that are closer to the lens compared to posterior tumors. So the oxygen effect. So as we know, nuclear DNA is usually considered to be the target of radiation. So when radiation is absorbed in cells, it results in DNA damage, such as single and double-stranded break that could lead to cell death. Ionizing radiation can act directly on its target, so directly on the DNA, or indirectly with other molecules such as water to form free radical and reactive oxygen species that can in turn attack the DNA. Then we have high versus low LET. So LET describing the amount of energy that deposited by radiation in a unit distance. High LET on the bottom have this dense tract and act mostly through direct action, while low LET are more sparse. So then top the most sparse, and most of its action, it's indirect. There are several factors that we know to change the damage caused by ionizing radiation on biological system. That we include dose rate, fractionation, the age or position of the cell in the cell cycle, the cell type, but also chemical sensitizer and desensitizer. So that will include reactive oxygen species scavengers, antioxidant. Remember that radiation will induce a high production of these reactive oxygen species. We also have drugs, and we have oxygen, which is going to be the focus of the rest of the presentation. So the oxygen effect was discovered was first described by Schwartz in 1912, when he saw that the skin reaction to a radium applicator is reduced when the applicator is pressed hard onto the skin, probably due to a decrease in blood flow, therefore a decrease in oxygen level. In 1921, we have Hartison, who observed that ascari eggs, which are a type of nematode worm, are more resistant to radiation when oxygen is absent. Petrie in 1923 showed a link between radiosensitivity of vegetable seeds and oxygen level. And Moultrie, Gray, and Reed in the 1930s were the first one to quantitatively measure the oxygen effect in both mammalian and plant cells. So the oxygen effect, it's the enhancement of cell killing in the presence of oxygen. The graph that you see on the left is the survival curve of mammalian cell exposed to X-ray with or without the presence of oxygen. So to generate such a graph, we grow a specific number of cells on a plate, we irradiate them, and then we grow them for we grow them some more and count the surviving fraction, which is the proportion of cells left with the ability to replicate and form colonies. And then we use a similar plot with dose on the x-axis and survival fraction on the y-axis to illustrate the cell survival after irradiation. And you can see that the survival curve in oxic, which means in normal oxygen level, has a steeper slope, which means lower dose of radiation required to achieve the cell killing oxic versus hypoxic condition meaning lower oxygen level. And that difference that you observe, it's what is called the oxygen effect. One explanation of the oxygen effect is the oxygen fixation hypothesis. It stipulates that the lesion caused to DNA by radiation are fixed and therefore harder to repair when oxygen is present, because you have the formation of this peroxide group, which are very stable and harder to repair by the cell repair mechanism compared to the radical form in the absence of oxygen. And for this to occur, you need oxygen to be present during or within milliseconds of the irradiation, since they act on radicals that are very short-lived. Another hypothesis for the oxygen effect that has been gaining attention is the mitochondrial stress explanation. And it considers mitochondria and not nuclear DNA to be the main target of radiation. So mitochondria, the figure on the top, as we know, are the powerhouse of the cell. They produce cellular energy, or ATP, by oxidative phosphorylation, which is illustrated in the bottom picture. So pretty much during respiration, glucose is broken down by a series of biochemical processes, which lead to the transfer of electron by different complexes in the inner membrane of the mitochondria, with the final acceptor being oxygen. And during this process, there is some normal but low leakage of electron, and this electron can generate reactive oxygen species that can damage the DNA and damage the cell. So radiation, especially low LET radiation, creates free radical from the radiolysis of water, which induce the release of calcium ion into the cytosol of the cells from the endoplasmic reticulum. And some of the calcium can enter the mitochondria and interfere with the electron transfer chain during aerobic aspiration, increasing the electron leak from the electron transfer chain, which boosts the production of reactive oxygen species. If too much of the reactive oxygen species are produced to overload the cell defense mechanisms, then you have mitophagy, which is the death of the mitochondria that occur. And if there's too much mitophagy in the cells, then cell death can follow. So we use the oxygen enhancement ratio, or OER, to quantify that effect, the oxygen effect. And the OER is pretty much the ratio of hypoxic to aerated doses needed for the same biological endpoint, needed to achieve the same biological endpoint. So the biological endpoint can be DNA damage, it can be cell survival, or it can be oxidative stress. And as you can see on the graph, it showed the cell survival curve at different doses. And if the biological endpoint is cell survival, then the OER will be the dose in hypoxia divided by the dose in normoxia to achieve a survival fraction of 0.01 in this case. And typical OER value for low LET such as X and gamma ray are between 2.5 and 3.5 at high dose. The oxygen effect is also observed at very low oxygen level. So as low as 0.001%, which is equivalent to 0.0075 millimeter mercury, as seen on the graph on the left. So if you look at this graph going towards toward the left, you have an increase in oxygen level, which increases the amount of cell death at a specific dose. And also the figure on the graph shows how the OER increases with oxygen level. So you have an initial but slow increase in OER value at low oxygen level, followed by a sharp increase in OER until it reaches saturation at high oxygen level. We also know that the OER, the oxygen enhancement ratio, is the highest for low LET, such as gamma ray, and lower for moderate LET radiation, such as neutron, and high energy alpha particle, but also they're almost inexistent in high LET alpha particle. If you see on that graph, on that curve on that picture on the right, so you have X-ray having the highest OER max, OER values, followed by deuterium, and then high energy alpha. And then for low energy alpha particle, the 2.5 mega electron volt, the curve is just a straight line, which is the OER is equal to 1. So no oxygen effect observed there. So what do we care about the oxygen effect and its role in its potential role in cataract formation? Well, the lens, it's considered to be the most hypoxic tissue in the body. With this picture shows the oxygen gradient and oxygen diffusion in the eye. So if you look at the blue arrows, it shows you how oxygen diffused across the eye tissue. And oxygen levels are the highest in the retina, the choroid, and the cornea. So 22 to 24 millimeters of mercury, or about 3% oxygen. And it diffuses through the aqueous humor and the vitreous humor to reach the lens. And oxygen levels at the lens nucleus is around 0.1%, which makes sense in order to prevent lens fiber cells rich in crystalline, they're very rich in crystalline, which are retained for life. So with aging, they accumulate damage to oxidation. So it's very important to protect them in order to lower the risk of damaging the lens. So therefore, we expect any risk factor, including ionizing radiation, that can change the oxygen tension in the lens or around the lens to cause oxidative stress and inflammation leading to cataract formation. So if you remember when I mentioned the irradiation of tissue around the lens, which could lead to cataract formation. So those tissue can be, so it can be the vitreous tumor gel just behind the lens, which is known to degenerate or liquefy due to irradiation, which makes it easier for oxygen to diffuse through it and get to the lens. And you also have the vascular retina, which is another possible source of oxygen. So for our project, the goal of the cataract project is to evaluate the oxygen effect, therefore calculating the OER values at different oxygen levels in LECs, in lens epithelial cells, when exposed to gamma ray, which we are hoping will help us understand how increase or decrease in oxygen level in the eye will impact these lens epithelial cells, leading to cataract formation. So we're using immortalized human lens epithelial cells, which have been modified with an SV40 viral sequence to allow us to culture them long enough to observe this effect. And we're using cobalt-60 as our gamma source. So for this project, we also acquire an oxygen chamber in our facilities. So it's an in vivo 1000 watt chamber hypoxic workstation, which is a live cell incubator, allowing us to accurately control the oxygen levels from 0.1% to 23%. We can also reach agnoxia, which is 0% oxygen by using pure nitrogen. We don't have that yet, but we're working with the vendor to set that up in our facility. The biological endpoints that we chose to calculate the OER are the colony forming units and the gamma HUX formation. So for the colony forming unit, we are measuring the clonogenic capability of LECs after irradiation, which means that the ability to divide after being irradiated, which is known to be affected by radiation. So it's the most common method used in the literature to calculate, to evaluate the oxygen effect. And for that, we seed the cells are low density on plate. Then we expose them to various oxygen conditions in the chamber. Then we irradiate the cell immediately with different dose of radiation. In this case, we use 2 and 5 gray, and we grow them for 2 weeks. And after 2 weeks, we count the colonies that were formed, and we can calculate the surviving fraction as a ratio of colony formed to number of cells seeded. So if there is no effect by radiation in theory, there should be as many colonies as cells seeded, plus or minus a certain percentage, depending on the plating efficiency of the assay. And we can then graph this survival curve and use that data to calculate the OER value. So for the gamma HUX assay, the formation of DNA double-stranded break after irradiation can be quantified using gamma HUX staining. So HUX is a histone protein attached to DNA to which a phosphate group is added at the side of double-stranded break as a result of exposure to radiation. So we can detect and measure these phosphorylated HUX protein using an anti-gamma HUX antibody and use flow cytometry to quantitatively measure them, from which we can generate those dose and effect curves and use those curves to calculate the OER value. So I'm only going to show you the gamma HUX as a result using to measure DNA double-stranded break because I'm still processing the colony forming unit assay. It takes a while to count those colonies, but as expected, if you look at the graph on the top, we saw a higher expression in gamma HUX at higher oxygen level, which means that more double-stranded breaks, more DNA double-stranded breaks are formed in hypoxic compared to aerated condition. So the same trend was observed for OER values with oxygen level, which is consistent with what we expected. We use this OER value calculated, and we fitted that data to the Howard, Flanders, and Alper equation for repetitive reaction. And it's a typical equation describing the shape of the oxygen curve. And based on that, on that curve, we estimated the OER max in LECs after exposure to gamma rays to be 3.8, and a half OER max happening around 0.6% of oxygen. So we definitely need more dose point and replicates in order to refine that curve. And another thing that is important to mention here was that the calculation were made while assigning 0.1% oxygen in OER value of 1, which is not usually the case. So usually in OER value of 1 is given to complete anoxia, which is 0% oxygen. We're not yet able to reach that in our chamber, but we're working to set that up. So what are the implications of that oxygen effect? So we determined the oxygen enhancement ratio, or OER, which describe the boost in cell killing after irradiation with gamma ray when oxygen is present, which means that the lens is very sensitive to oxygen changes, and having low oxygen level makes the lens well protected by the oxygen effect against low LET, not so much high LETs radiation, but more low LETs. So therefore, we are expecting that any slight changes in the distribution of oxygen around or at the lens could cause severe damage upon radiation exposure, which explains the prevalence of cataract in patients in higher or lower than normal oxygen level in the eyes as a result of disease such as diabetes. So future direction, we are working on refining the oxygen curve by adding more replicates and those to address some of the variation that we've seen in our data. We're working on installing the anoxia mode in the chamber to reach 0% oxygen. And we would also like to determine the oxygen enhancement ratio, so evaluate the oxygen effect at lower dose, so less than 1 gray. So the oxygen effect are typically lower at lower dose compared to higher dose. So we would like to investigate that in LECs. And we also would like to look a little bit more into the biological mechanisms of oxygen effect in lens epithelial cells. So we want to understand why low or higher than normal oxygen levels in the eye can induce cataract. So we want to know what the role of mitochondria and the production of reactive oxygen species, as we know that irradiation increases the formation of these reactive oxygen species, and see if these levels are affected by the oxygen levels, the oxygen concentration. We want to understand the role of HIF proteins. So HIF proteins are hypoxia-inducible factors, and these factors are produced by the cells when exposed to low oxygen level. So they have an effect. They can affect many cellular processes, including cell division, metabolism, and gene expression. And we want to see if there is a link between HIF protein induction and genes involved in the cell radiation response. We would also like to look into the epithelial to mesenchymal transition of LECs, of less epithelial cells, as well as lens fiber cells, as a result of radiation exposure, which is a proposed mechanism, as I mentioned before, for posterior sub-capsular cataract. So when we have that EMT transition occurring, the cell will lose the epithelial markers and gain in EMT markers. So we can analyze the loss of epithelial markers such as ZO1, which is a tight junction protein, or E-cadherin, or beta-catenin that are involved in cell-to-cell adhesion, as well as the induction of known EMT markers such as Notch1 and alpha-smooth muscle actin. So in summary, report of in vivo study of whole lens and lens explant show that the lens is one of the most radiosensitive tissue in the body, which is paradoxal because we will expect its extreme hypoxic nature to shield it from low LET radiation by the oxygen effect. So we did confirm the oxygen effect in LEC with an OER max to be a high dose to be 3.8. Therefore, we will expect any changes in oxygen at or around the lens to change the sensitivity of lens epithelial cells to gamma irradiation. So we are not aware of a study using hypoxic condition to investigate radiation-induced cataract, but we know that oxygen level can have a significant impact on cellular mechanisms. So it will be interesting to understand the dynamic that is going on here. So we have ongoing experiment to refine the oxygen curve and further our knowledge of the oxygen effect in NSS and its role in radiation-induced cataract. So with that, I would like to thank my other fellow CNL workers that contributed or that have contributed or still contributing to this project. So it will be Mega, Tyler, Yi, and Mike Rollins. And also mention that this work was supported by the Federal Nuclear Science and Technology Work Plan of Atomic Energy of Canada Limited, so AUCL.